Previously, we collected our environmental DNA samples in Bristol Bay, Alaska. So, now what? Back in the lab at the Alaska SeaLife Center, our scientists are preparing the samples for DNA extraction. The water samples are poured through a funnel onto a special filter paper, which catches the molecular material as the water is vacuumed out. It's a rather simple process. However, it does involve some patience. Once all the water has been siphoned through, the filter paper can be removed. The paper is cut into tiny pieces and then placed into a tube. The tube is extensively shaken, extracting the remaining liquid carrying the DNA to the bottom of the tube. After a purification process, it is run through quantitative polymerase chain reaction, or qPCR, to amplify the DNA contained in the liquid. We observed all kinds of life around our sampling sites, so how can we possibly detect DNA presence from just our targeted species? We used a specialized test kit, allowing us to compare a DNA sequence unique to our targeted species with the extracted DNA in order to find a match.
If this does prove to be successfully validated, we would want to take it to Bristol Bay and collect samples and work there. But then ultimately the idea is to pair what's available with what's actual walrus movement patterns and look at covariates to all of that in this centralized location. So Bristol Bay is a good place to study for that. There's a lot of local knowledge, a lot of traditional knowledge that we can pull from, and there's a lot of agencies and support out there that will be really helpful in kind of moving all that forward.
That pretty much wraps up our primary objective for this first pilot study that was funded through the Dr. Holly Reed Conservation Fund, which is through Point Defiance Zoo. So we're really excited to kind of talk to our partners there and other walrus researchers around Alaska and in the Arctic and start to see if there are maybe other questions that we can answer to improve how eDNA can be helpful in the field. Like we mentioned, looking at some of these experimental studies to see if we can get a better idea of prey composition, species composition, or residency times, so we actually have a tool that we can use and transfer into the wild.
We have learned how environmental DNA can be a non-invasive tool to help monitor our ecosystems, such as detecting the presence of key prey species. Continued monitoring of Pacific walrus prey abundance may allude to drivers behind walrus movement patterns at and between terrestrial haul-out sites. As we continue to grow in our understanding of eDNA, we may use it across a variety of studies and learn how to better monitor and protect marine environments, all from within a few drops of water.